So today I'm going to talk about microbes, sex manipulating microbes in insects. So firstly, I'm going to talk a little bit about my institute. So this is under the Ministry of Agriculture, and our institute is called NARO, National Agriculture and Food Research Organization, which consists of 20 centers and institutes. So, and one of them, Institute of Agrobiological Sciences, is the institute that also focus on pure science and also the applied sciences to apply to agriculture. And our research unit is Institute Microbe Research Unit. We are investigating the interaction between insects and microbes. So today I'm going to talk about the bacteria and other microorganisms. As many of you know that the microorganisms some of the microorganisms are free living, but many others are very deeply associated with other organisms, typically with eukaryotes, many different kinds of eukaryotes. And broadly, they can be classified into two types on the basis of transmission route. First is a horizontal transmission that are often called infectious microorganisms. And they are either directly to person or organism to organisms. And sometimes in other cases, they have an intermediate organism to transfer the microbes into other organisms. And the second one is vertical transmission. Vertical transmission is it's called heritable microorganisms that are transmitted from parent to offspring. Particularly in arthropods, including insects, the female typically transmitted the microorganism to offspring because some of them have variety of microorganisms inside the cells. As you can see here, this is a cell, insect cell culture, and that has a nucleus in the center, and you can see a lot of small dots around here. This is the bacterium called Wolbachia. So Wolbachia can be transmitted to next generation via female ovary. And this is
okay.
Can you hear me?
Yeah. Sorry to interrupt. Can you, at the bottom you see a tab there, one bar is coming. Can you hide it? Yeah.
Sorry. No. This one, you mean?
Yes. So there is a hide. Somewhere it's written hide. You can click that, it will disappear from the...
It doesn't disappear.
No.
Okay. It's
so
okay now.
Okay, it's fine now. Fine.
Fine. Okay. Sorry. And the photos on right side is electron microscopy. This is called another different bacteria called the Wabachin and spiroplasma in the insects within the insect cells. Such symbionts, we call this endosymbionts or bacterial symbionts. These can be considered as the genetic elements of the host organisms. Sometimes it's called holobiont. Holobiont is the host, and all of its symbiotic microbes are called together as and considered as the unit of selection. So we can consider it's a kind of genetic elements. So now I'm going to talk a little bit about the surface genetic elements. Surface genetic elements, it's genetic elements that can enhance their own transmission at the expense of other genes in the genome, even this has no positive or negative effects on organisms. This idea is first explicitly stated in Nature in the same issue in 1980. And after that, the examples increased, and you can see a lot of such kind of examples. And you can see at the bottom, cytoplasmic sex ratio distortions, which include variety of microorganisms, which has been found in many different species of insects. And there are many very excellent review papers are published to date. Yeah. So as you can see, why such kind of surface genetic elements arise is based on the because there is a existence of conflict, because not all the genes are inherited in the same manner. Some, as you can see here, some genes are maternally inherited, and other genes are paternally inherited, and the others are biparentally inherited. And they, for example, for cytoplasmic parasite, is more advantage to bias the sex ratio toward females, because female can only transmit the element to the next generation. So such kind of examples and case studies are summarized in this book, Genes and Conflicts, written by Bart and Chivers. And I'm explaining about the cytoplasmic parasite that living inside the cells of inside the cell. And they are only transmitted from mothers, as you can see, to their offspring, males and females. But the cytoplasmic parasite in males have no transmission route anymore. If the host dies, they die together. It's a dead end. So biasing the host sex ratio towards females is advantageous manipulation strategy for the symbiont, and actually do. And there are 3 types of distortions. All of them causes a female biased sex ratio. And another host manipulation is called cytoplasmic incompatibility. This is a very tactic behavior of the microbes that microbes in the mating between non-infected mothers and infected father will produce unviable offspring. They do not grow. They die at the embryonic stage. And the mechanism is not well understood, but the infected males do some kind of genomic modification to kill all the offspring. But in the infected, and the female mother infected, if the microbes are inherited, they can rescue the modification so that they can increase their proportion among the population rapidly. So this is called cytoplasmic incompatibility, and the others are female bisex ratio distortion. And the causal agent, you can see there are many different kinds of bacteria, but you can see the Wolbachia, this rickettsia-like bacteria are found from every categories. And another one is cadenium. This is quite distantly related to Wolbachia taxonomically, but they cause 3 different phenotypes. And also you can see rickettsia. And so you can see the bacteria are quite different, but they cause similar phenotype. Yeah. So So you can predict that if the bacteria can change the sex ratio toward bias sex ratio toward females, and after generations, the population sex ratio will bias toward females, and because they need males to produce offspring, if the too much biasing toward females will make the host population go extinct. It's a prediction, but in a butterfly study, in a butterfly has revealed so nuclear resistance we call suppressor is easy to evolve among host genomes. As you can see, this butterfly is famous for insect collectors and from all times. And in the early 1900s, this butterfly population in South Pacific islands called Samoan Island, only less than 1% of the population are males, and sex ratio is extremely female biased. And this female biased sex ratio was also observed in 2000. Yeah, but surprisingly, after 5 years, they found sex ratio returned to completely normal. So the detailed crossing experiment using butterflies has revealed that host genetic change has resulted in normal sex ratio, even though Wolbachia bacteria retain the male killing ability. So it was such kind of host resistance was theoretically predicted in all times. This is the first empirical case to demonstrate this phenomenon. This and after that, this is a similar phenomenon has been found in spiroplasma-induced mercury. This is called the different bacteria, but the similar rapid change of the sex ratio, population sex ratio, has been evolved in 5 years. Yeah, this is a study that I've involved in. And in Japan, the sex ratio was quite biased toward females, not so extremely biased like the case of the butterfly, but anyway, males are around 10%, but in 2016, males become 40%. But we had the breeding stock of 2000 breeding line of 2011, so we could make a crossing experiment to see whether sex ratio restoration was the cause of the host suppressors or not, and it is actually the case. So I was lucky. We were lucky enough to see that because we had the previous data in 2011. If we started the experiment in later than 2016, we would have never observed such kind of male killing or anything. Yeah, if you collect, last year we also collected many lacewing from around Japan, and all of them, many of them are infected by the spiroplasma, but they produce 1:1 sex ratio, consistently produce 1:1 sex ratio. So we can guess, because we have all the data, we can say such kind of dramatic change have happened. So similar thing would have occurred in many other different species of insects and other different bacteria combinations. So this is things that we cannot predict beforehand. But if you move the Wolbachia into a different host genetic background, we may be able to guess what happened in the past. For example, this is an old paper made by Professor Sasaki in Tamagawa University. He was at the time, he was an assistant professor of the University of Tokyo, and he moved the Wabachin strain WCA from naturally infected Quadricautila into closely related different host species. And in natural species, they do not cause male-killing. But after moving to the different species, they induce male-killing. So you can guess in Quadricautila, in a whole species, they have already evolved a suppressor against male-killing, and their suppressing gene has fixed in a species. But if it's the Cuniella, because they don't have any such kind of Wolbachia in nature, they don't have any suppressors, so that we could observe such phenotype. But after that is John Yannick. He's a professor of the University of Rochester. He also found a similar case in the Drosophila. He moved the Wolbachia by backcrossing, because the species are very closely related. They can make hybrids by crossing. So such kind of phenomena has repeatedly and frequently may occur in nature. So we can guess if you find the host symbiont relationship, and yeah, for example, if there's some kind of insects had a particular symbiont, such as Wolbachia. Wolbachia is quite common, and you can see, and you don't see any phenotype, but you cannot trust it. Really, symbionts may have you can look back, and symbiont may have acquired killing or other phenotype, and host may have acquired the suppressor against it. And after that, the symbionts may have another phenotype, and the host may have suppressors. Again. So we don't know how many of accumulated in the genome. So it's called a cytonuclear arms race, but it may happen in nature. We don't know how common it is. But as you can see here, the sex determining system is very diverse among insects. And even in a closely related species, you can, even the sex determining genes are different. And even in a chromosomal level, some species have X, Y systems, and other are the other way around, Z, W, ZZ, or some other species even don't have any 6 chromosomes, and they are produced by sex determined by environment, or haplodiploid sex determination systems, and pathanogenome elimination, and so many type of systems. For example, we can imagine that such kind of endosymbiosis by bacteria can contribute to the diversity of sex determining systems. I don't know how common, but in some cases it should be. For example, in a woodlouse, and woodlouse sex determination is research has long history by the French team in University of Poitiers from the 1960s or 70s. They produce, they make sex determination by female heterogametic sex chromosomes. Like this, female has ZW and male has ZZ. It's called chromosomal sexual determination. But in Wolbachia infected female have the ZZ chromosome. These are genetically male. They become, but the presence of Wolbachia made them female. And by mating with the ZZ males, they produce only ZZ offspring. So sometimes they lose the Wolbachia, then they become males. So it's a kind of a cytoplasmic sex determination. Wolbachia decide whether they become males or females. And after that, there's some interesting cases that the autosomal gene that determine as a male. So it's a newly acquired sex chromosomes. Once it used to be a autosomes, but the sex chromosome, ZZ chromosome, are not sex chromosome anymore. All of them become ZZ. So they behave like our autosomes. So you can see it's a kind of XY system. So by changing the sex determination to cytoplasmic sex determination, they change the system from ZZ and XY system. And they also found a different type of female determining element called F element. This was found to be actually a Wolbachia genome integrated into the host chromosome. And they also found that that once it was an autosome, but now they can be considered as the sex chromosomes. And ZZ behaves like autosomes. Yeah, it's a quite new study. And similar to that, we have also found ZW and ZZ sex determining system has interrupted by Wolbachia. So in this moth, Wolbachia infected females can be found from nature in about 5% in frequency, and the infected female produce only females. But the ZZ individuals die because of Wolbachia. But interestingly, the Wolbachia determine the sex at the very early stages of development, but by examining the sex-specific sex determining gene, we can see that they are female determined, but they die. But if you cure the Wolbachia by antibiotics, they produce only males by killing the WZ female, WZ male individual. But actually they are determined as males. So it's quite complicated, but we can say now that the W has a female determining function in normal individuals. But Wolbachia has ability to determine its host as females. At the same time, the double chromosome in infected line, the double chromosome did not have female determining function anymore, and Wolbachia is compensating for it. So but in cured line, they cannot determine as females, and they become males, and they cannot survive because of the dosage compensation dosage compensation is a mechanism to balance the G-linked gene dose. So if they are determined as males, the two Z chromosome is too much for them and decrease their gene dose to half, and they keep it as a 1. But in the case of Wolbachia infected these individuals, they're determined as females and do not dosage compensation do not work properly, and their expression is too much and they die. Actually, this was experimentally shown by quantitative PCR and RNA sequencing. On the other hand, in the cured line, the males, so although they have only one Z chromosome, they are determined as males, and they are, by the function of dosage compensation, they are regulated by 1/2, and they are too small and they die. So that's all we looked, but it's quite a different story from the woodlouse. But in Lepidoptera species such as moths and butterflies, Z and W chromosomes, Z chromosome are so, Z and W are so differentiated, and dosage compensation is very strict. So we can see here that this is what we have observed. Wolbachia play a role as a female determiner, and W chromosome has lost it. But in between the normal cases and the Wolbachia in Australia case, there must have been something in between. In this case, the W and Wolbachia so we call it a normal making phenomenon, widely observed in different species of insects. So it's a kind of redundant state, and spontaneous loss from double chromosome can occur. So it's a kind of speculation. And after that, we speculate that double chromosome do not have any function anymore, because we know that the double chromosome are full of repetitive sequencing and transposons, and no functional genes are known to date. So it's possible to speculate that the all sex chromosome may evolve from this. Actually, we found such kind of similar case in another species of butterfly. In this species, we can say that there are two different kinds of Wolbachia here, but the Wolbachia called WCI is fixed in the species, but they do not cause all female or affect any sex ratios. And WFEM cause all females. But in Tanegashima Island, this is my field site. The sex ratio, so we started observation from 2005. The sex ratio is quite female biased, but it fluctuate, but they do not go extinct, and no suppressor evolved until now. But they are somehow maintained in the population, but I don't know why, what is happening. But the graph on the bottom, you can see this is a proportion among females, whether they are double infected. I mean the all female producing ones or non-normal females. So the trends are quite it's quite similar to the sex ratio. And we can, so there are two type of females. One is single infected, and the other females are double infected. And the chromosome observation clearly show that the normal females are DW and the WFM positive females are DO. It's a quite different sex chromosome constitution. And we can, when we observe the embryo and eggs, in normal females, they produce both DW and ZZ, and but WFM positive female produce only ZO embryos. They do not cause male killing. Yeah. So, but if we cure the adults, they start to produce also ZZ. So it's clear that the Wolbachia disrupt the maternal inheritance of Z chromosomes. But the detailed mechanism is still unknown. There are, 3 possible mechanisms, but until now we cannot discriminate these. So we can see here that this is also a hypothetical evolutionary trajectory. That is this is the one we shown the previous in the previous case, and this is what we have found. Wolbachia has lost, double chromosome have been lost, and Wolbachia may have acquired a new function to disrupt maternal inheritance of Z chromosome. So as you can see that the sex determining system is diverse. Diverse system can be evolved. This may partly be explained by endosymbiont. Yeah, that's sorry. My explanation is a bit complicated, but it's also the Wolbachia can cause many different effects on biology of its host, such as speciation. Not only sex determination, they can cause speciation by accumulating their cytonuclear arms race. So it's a kind of interesting model to examine evolutionary biology. And if you're interested, you can check my homepage and write me email. Okay, so that's all my talk. If you have questions, please ask me anytime.
So thank you, Professor Daisuke. That was a really nice talk, insightful. So right now we welcome questions. You can unmute yourself and switch on your video, and you can get your time to interact with Professor Daisuke. And if you face any technical issues with internet, please feel free to write your queries in the chat box. Yeah, you can ask questions to him.
Hello, Darshan.
Hello.
Yeah, Ranjit here.
Oh.
Yeah. So I had a doubt on the Urema species.
Yes.
Yeah. So you found that in some cases it was like all female populations, but in some you had both male and female sexes. You were saying, right? Even though Wolbachia was present there in your initial slides. Can you hear me, Raji?
Oh, you mean the
Just a minute. I think there is a noise coming from there.
Just a second. Yeah, please mute yourself. Yeah, please.
Yeah, Daisuke. So I think, is it that in Urema species, So is it that some particular population has actually you have all female biased, and some other population where it is sex ratio is more or less equal, even though same strain of Wolbachia was present? Is it like that, or is it actually different strain of Wolbachia were there?
Oh, yeah. In the normal population, in other populations, there are no W frame strain. They are fixed with a single strain of Wolbachia called WCI. They call CI, but they do not affect sex ratios.
Sex ratio.
Yeah. So we all of them produce 1:1 sex ratios.
Hello.
Hello?
Can I ask a question?
Oh, yes.
Sir, can you hear me?
Oh, yes. But
Okay. Yeah. Now, of late, there has been a lot of interest in controlling mosquito populations using Wolbachia. There has been a certain amount of success in countries like Vietnam and Australia. Has there been any efforts in this direction to control mosquito populations in Japan? If so, did you encounter any problems related to Wolbachia infections to other species other than the mosquitoes? You got my question?
oh, do you mean that Wolbachia can be moved into different species of insects, right?
See, basically, they use this property of cytoplasmic incompatibility to sterilize these mosquitoes in malaria control. Okay.
Yes.
My question is this: can this Wolbachia present in mosquitoes be passed on to any other species? For instance, those predators which feed on the mosquitoes, can they incorporate Wolbachia?
I think, hello, Daisuke.
Yeah.
Yeah, so I think Professor Rao is asking something. It's like because the strain which is present in mosquito control, okay, so through the ecological interaction, let's say through predation, can that strain be transferred to a natural predator? And whether a new predator in the natural population can get infected by the Wolbachia which is present in mosquito that we have introduced into the nature.
Yes, it cannot. Yeah, this possibility cannot be completely eliminated, but it should be very, very rare. But we cannot say it cannot happen. Yeah.
So I just to
excuse me. For instance, you have worked on some insects, sex determination and things like this under Wolbachia influence. Suppose those insects have been fed upon by, say, a bird or some other predator. Would Wolbachia be passed on from the insect to the predator?
Yeah. So in evolutionary time scale, they can be transmitted by, for example, the parasitic wasps and so on to different species. But in we mean the ecological time scale, if you if you pick up a single species of insects, we always find a single species of Wolbachia. The Wolbachia strains are fixed, depend on insect species. So the Wolbachia cannot freely move between different host species. But from an evolutionary viewpoint, it must have occurred in, for example, in 1 million years, once in 1 million years or so. So, yeah. Do you make sense?
Yeah. So, Professor Daisuke, what I was asking is that the current knowledge of Wolbachia spread is actually limited to arthropods and few species in crustaceans, right? So we have very few information. I mean, we think we have no information that Wolbachia actually can reside and infect the cells of any other taxa. I mean, outside the insects or crustaceans, we have probably very few. So I think predators, whether they will get established in such a predator or not, that itself is a question, right?
Can I ask you a related question? Now, can you speculate the evolutionary reasons why Wolbachia parasitization has been confined mostly to insects and a few crustacea and not even any other invertebrates? Are there any barriers for Wolbachia?
I mean, why they are limited to only insects and crustaceans? Why not other taxa?
Hi. You mean the other taxa than arthropods?
Yeah.
Yeah, so it was at least in some nematodes, filarial nematodes, Wolbachia has been found from many species, but the Wolbachia lineage is different. So yeah, we can call them a different species of Wolbachia, even though the species are not named yet.
But does this taxa have something specific that why they are only found in these arthropods and nematodes, not in any other related groups?
So yeah, we have
that is
no idea, but somebody examined for surveyed mollusks at they examined about 300 species, but none of them are infected. Yeah, so we don't know what limit the boundary of hosts.
right.
Can you experimentally parasitize any invertebrate with Wolbachia in the lab?
Yeah, among arthropods it's possible, but we don't know how far it can infect. But I think it's quite difficult to infect non-arthropod species. But not many people have ever tried. I have never tried in other non-arthropod species. Yeah.
Thank you.
Thank you very much.
So we have a question in the chat box. If you can just answer that would be nice. From Astosh, there is a question in the chat box. If you can see that, is there any benefit for the microbe in determining the sex of its host?
Benefit.
So Daisuke, he is asking that because the microbe or the Wolbachia.
Oh, sorry. I saw it now. I see it now. Yeah, it's a great advantage because the female is very important for microbes because only females can transmit the microbes to next generation. Because so if the hosts are determined as males, they lose their destination. So it's a great advantage to determine its host sex. Yeah, it's okay.
Hi, sir. I have two questions. For the lacewing population, you told that earlier in 2011 there was a Wolbachia infected population, and after
Mhm.
that another population evolved, which around 2016, I think, that which are suppressed Wolbachia male
Mhm.
determination.
Yes.
But how did then you told that you also kept the 2011 strain so you could cross them, the 2011 strain and the newer strain, but how you kept the 2011 strain, how it survived in such a female biased environment, and what was the dynamics of that population?
Uh, in the lab, we maintain the 2011 line, but many of the 2011 lines are infected by the spike plasma, and they only females. But so we cured the bacteria by antibiotics, and they start to produce 1:1:6 ratio, and we maintain the laboratory line for about 5 years. And so the 2011 line don't have a suppressor against male killers, but the 2016 the suppressor has already almost fixed in the population, and we can, by making the back crosses, we replace the host genetic background to the 2011 background, keeping the spread plasma alive. Then
Okay,
the
thanks.
spread plasma begin to produce only females. Okay. Does it-
Okay, thanks for that. And second question is that you told that Wolbachia can like the speculation, the evolutionary speculation. You told that from the chromosome some part can be deleted, which can be complemented by the Wolbachial genome. So can that be occur by some RNA silencing or RNA interference processes instead of gene deletion kind of that, because these are more easily accomplished?
Well, oh, sorry. What is the question?
So I asked that you told in the evolutionary speculation of the population that the FIM gene got somehow deleted from the W genome, and which was complemented by the Wolbachia.
Yeah.
So, is it really necessary that the film gene would have been deleted, but maybe it may happen that the Wolbachia may suppress that gene by RNA interference or gene silencing?
Um, but you may have.
Do we have the sequence of that W gene? I mean
No,
W chromosome?
no,
No,
not
right?
at all. It's just a
That's
yeah, it's just a
right, right.
speculation.
Yeah, yeah. So it could be
Mm,
that what Harish was asking, that it could be that it actually deleted from the W chromosome, or it might be it is there, but it is getting suppressed by somehow uh, by the Wolbachia genome. Maybe Wolbachia genome has some part which is suppressing the
mhm.
FAME gene in the W genome.
Yeah,
I mean,
yeah, we are sequencing, and we are trying to see, but
yeah, yeah.
the double chromosome is very difficult to sequence. It's
Because
almost
of
like
repetitive regions.
it's like a junk sequence, and so Yeah, but still, if you cure complete, if we can cure the Wolbachia completely from the line, so we can, but then they produce only males. So that is the base
Yes,
of
yes.
yeah, base of
Yeah, I think this somehow answer this question, because if we remove completely the Wolbachia genome, then if the FAME gene is still there with a W chromosome, then it should become a female, right? But
Yeah.
it is not what is happening. So that
Yeah.
means it is not that the Wolbachia is actually suppressing it. Probably a FAME gene is not there in the W chromosome.
No, but if suppose the Wolbachia genome is not there, so the Wolbachia may not be producing some interfering RNA which would be suppressing the host genome. So therefore, when Wolbachia is removed, then the host can return back to its normal sex determination.
But then every generation, let's say you have a next generation where you don't have Wolbachia at all, then I mean, from its embryonic stage itself, and if you don't have the FAME genome, then female determining factor is not there, so female won't be produced. Right? Because for the silencing effect, Wolbachia's present is somehow necessary. Otherwise, some part of Wolbachia genome must be integrated into the host genome.
Yeah.
Then only it can work, right?
Yes.
Yeah. So if it is integrated, then that's one speculation, right? But if it is not integrated, then Wolbachia presence is necessary.
But they showed that after treating with tetracycline, so the normal sex determination returned
But it is
in many.
not in the same generation. It is like next generation.
Okay, yeah. So
Yeah, it's not that same you took a male and treated it became a female. It's not like that. It's the next generation because it transmits through egg. So when you treat it, you see that in the egg stage you don't have Wolbachia. So what you will see the effect in the next generation.
but if the genome was completely removed, then it should, I mean, say particular part of the genome was removed from the gene, then I mean from the chromosome, then it should have some effect also after treating with tetracycline after in the next generation and other generations too.
So I think we need a sequencing or somehow targeting
Yeah.
the FAME gene so that we can know that actually FAME gene is physically present or not. Right, Aishuke?
Yeah, but it's a bit complicated. Yeah, I'm not really sure. It may have some genomic imprinting. If the
Right.
Wolbachia has been eliminated, it can have some may have some effect to men on the genome. So that, but it's but experimentally it's outside the scope of my work. But
Yeah,
it's
currently
good
it's
to
a
examine.
speculation.
Yeah, but it's good to examine if it is technically feasible. Yes.
Right.
Thank you, sir.
Yeah, the last the question. Go back to the question in the chat box. I see can Wolbachia infection affect mitochondrial genome evolution? Yeah, it's such kind of work has been done in many papers. So you can check the database. Yeah, actually, Wolbachia has a quite a profound, quite a big effect on mitochondrial genome evolution. It can decrease the diversity of mitochondrial genomes. And the next question, can you please explain the complex explanation to us? How it survives? So because of the formation suppressor. Yeah, it's kind of suppressor we can name of. The WM. It's a kind of it's a very classical genetic study made in 1992 or 3 in paper Journal Genetics. So no molecular work has been done, but but making a lot of crossing experiments, they clearly formulated that male determining gene generated in the autosomes has determined its sex, so it behaves like a sex chromosome. Yeah. So all the sex chromosome, Z chromosome are they have lost the double chromosome from the population, so it behaves as if it's autosomes. But the newly arisen sex chromosome, so they call it the N, behaves as if it's a sex chromosome. So it's a very interesting idea. Yeah. So it changed the idea of how the sex is determined. The sex determining system can change quickly. Yeah. Is it, I'm answering the question. Okay.
So, Daisuke, there is one more question. So in the chart works, and it asking that about your lace swing work in 2011 and 2016. And so it's asking that in 2011 you started the lab population, and then you 2016 you got that you collected new populations from nature, from wild. Then you backcross them, right, to transfer the suppressor gene probably, or the Wolbachia into the 2011 genome.
Yes.
Right. So the question Prachiti is asking is that when you grow this lab population, the genome itself probably have changed in the lab population. So I think, but see, I mean, I don't know, but I think from this is, can you see it on your chart box?
Yes. Oh, wait, just a moment.
Yeah.
Can you present it? Preserve the from earlier the backwards during the create image change in this time period. Oh, yeah, there might be some change, but as far as we can see from the data, we don't have any suppressor gene in 2011 line.
So it is
So
like we are. So is it like we are actually comparing the 2011 genome, which has probably we expect there is no suppressor.
Yeah, we cannot say it is exactly the same. We cannot say it's exactly the same, but we are keeping it without any sex ratio distortion or any selective pressure was present on the laboratory line. So there are no reason to consider that there are any change in the genetic composition.
So basically it is that are we comparing the differences like we are getting in the 2011 strains and the strains you are collected now recently in 2016?
So it's a at least in a suppressing genomic region, there must be a clear difference in 2011 and 2019.
Daisuke, can I just answer her?
Yeah.
Yeah. So I think Prachiti, it's like we are not comparing the strains. We are actually taking the strain Wolbachia, which is almost the same strain. I mean, because from 2011 and 2016, you can expect it is the same strain. But what we are comparing is that how does the strain behave? Can the strain have its own phenotypic effect, which is probably feminization effect, in the two genomic background? One genomic background of host, which is from 2011, and one genomic background of the host from 2016. Okay. So we are
Okay.
putting the Wolbachia in two genome and seeing that whether whichever the genome has the suppressor capability, which suppresses the effect of Wolbachia.
Okay. Thank you.
Thank you.
So we got a question from Anish in the chat box. Can you see that?
Yeah, the male or female bird population adopt any counter-evolutionary strategies to bring back sex ratio to 1:1.
So whether it is that male starts mating more frequently, let's say for a strategy, having this wild case.
Yeah, it's
Because in case of let's say the butterfly, that egg flies which we saw, hypolimnus.
yeah.
So we initially see it was almost 1% of the population was male, but then in 5 years we had a report that it is 1:1 population. But that is because of probably suppressor evolution of the suppressor gene. But how does the population actually exist, even with such a small male numbers? Even though there are very few male, how does the population survive? Why, why it doesn't cross, doesn't go extinct? Does the male have some adaptation? Let's say mating frequency increases, or female has some adaptation so that they are still able to sustain the population, even though population has very few males.
Yeah, so yeah, they can male behavior can evolve.
But do we have any particular study actually which address such questions? That in a biased sex ratio, let's say particularly by Wolbachia-induced sex ratio, whether the female has some evolved strategies which is linked to maintaining the population over a period of time.
Yeah, it's
Don't know. Maybe some study was there on the male mating strategy, like mating frequency and something. Very few were there. Don't remember anything else people have actually explored. This is probably something
Actually,
yeah, Professor.
Ranjit,
Yes.
I was also curious if there is any difference in the behavior of these parasitized versus non-parasitized females, reproductive behavior or any other type of behavior.
Right.
Yeah, but yeah, in a case we are working on in a butterfly, yellow butterfly, Urema butterfly in Tanegashima Island, we have just recently published a paper on bioarchive. In that paper, we examine the sex ratio. Female bias sex ratio is maintained for about more than 10 years. Although they are fluctuating, it looks like it's a frequency-dependent selection. It's very difficult to discuss, but it's likely to be stable. But it's not dependent on suppressors. There are no suppressors at all.
Right.
But the male may adapt to the sex bias. It seems likely that the male change behavior according to the context, or possibly by genetically determined changes, or they may plastic response to the surrounding sex ratios. And yeah, there are 2 or 3 possibilities which are explained in the discussion section of the paper. But it may be interesting to look in detail how whether by breeding experiment we can test whether males respond to the environment where they're genetically changed or not.
Okay. I'm still curious because this is something that you could study experimentally in the lab. You could take females, parasitized, but one of them could be, say, a group could be subjected to, say, tetracycline, kill all the microbes. And so you have a batch which has the microbe and the other one which is free of it. I'm curious if there is any changes in the reproductive behavior between these two, number one. Number two, would there be any difference in the number of eggs they produce? And things like that, just
Yes, yes.
and also longevity of the insect.
Yeah. Sometimes the breeding experiment has some technical hurdles, and for example, the antibiotics may affect insect behaviors, and
Yeah,
so many controls are necessary, and sometimes very difficult. But anyway, we are planning to do some kind of work. Maybe next year. Yeah. Thank you very much.
okay, thank you.
Gokul?
Yes. So it's time to conclude the session. Why we conclude today's session, and we are very happy to have Professor Daisuke with us. It was wonderful to have you in this series. Thank you once again, Dr. Ranjit, everyone, every participants, Professor, everyone. Thank you so much. We'll see you in next.
Arigatou gozaimasu.
どうもありがとうございます。
Thank you.